So we're going to introduce the idea of the endocrine system first. And when we talk about the endocrine system, I want you to be able to, in your head, contrast the endocrine system from the exocrine glands that we talked about before. When you hear exocrine, remember that those are ducted glands, and the secretion from the secretory epithelium goes out under the surface, usually of the skin. What we're going to talk about now is the release of hormones, or the release of molecules, the secretion of molecules into the extracellular space. Not through ducts, mostly through exocytosis, out into the extracellular environment. And then what we have to think about is this interaction between the secreted molecule and other cells. We'll walk our way through this in 3 steps. The first one is something known as autocrine. Auto meaning self. In the autocrine response, the cell that releases the hormone binds to the hormone and responds to the hormone. In other words, that cell that releases the hormone has a receptor for it. In a related arrangement, there's the so-called paracrine response. In the paracrine response, all this means is that the cell that's responding to the hormone is located nearby to the cell that's releasing the hormone. Para, in the neighborhood, nearby. And then the final one, and this is the one we normally think about when we talk about endocrinology or the endocrine responses, is the effect of hormones over a long distance. By and large, this whole section that we're going to talk about, and much of the whole lecture today, revolves around the idea of long-distance communication among cells. You have something like 1 trillion cells in your body, and they're molecularly far, far away from one another. And yet there can be remarkable communication among all those cells. And much of that chemical communication is carried out by hormones and receptors. So this idea of hormones and receptors will walk through autocrine, paracrine, and endocrine. Now, when we talk about the signal that's released from one cell and communicating to another, there are a handful of molecules we use for this. We can bundle them, really, into 4 groups. The first one we'll talk about is peptide or protein hormones. Whenever you see this term, peptide or protein, again, remember, it's a sequence of amino acids. It is a protein or a small protein, a peptide, that obeys all the rules we talked about for proteins. By and large, these are big molecules. They're charged. They're hydrophilic. They're water soluble. The other big group of hormones are amino acids, modified amino acids. But there are only 2 amino acids. Among the 20 amino acids we have, there are only 2 that we modify for hormones. One is a tyrosine, and the other is tryptophan. Tyrosine gets modified a couple of ways, and we'll talk about this briefly. The tyrosine that we're going to talk about almost entirely in this course is going to be the tyrosine that's modified into catecholamines. Those that are going to be dopamine or epinephrine or norepinephrine. The so-called catecholamines, really important in the nervous system. The other tyrosine derivative is really quite different. It's an iodinated tyrosine. It's thyroid hormone. It's hydrophobic, and it's produced only by the thyroid gland. And so we're not going to spend much time on that. Even though if you take an endocrinology course, you're going to spend a lot of time on thyroid hormone because it's so, so important for metabolic function. The other modified amino acid is tryptophan. And what tryptophan gets modified into is serotonin, often known as 5-hydroxytryptamine. Again, this is important in the central nervous system. It's also important in blood clotting and a number of other things, but it's really a modified amino acid acting as a hormone. Now, most of the time when we think about hormones, the first thing that comes to mind are steroid hormones. The one thing I want you to hold on to about steroid hormones is they're a modified cholesterol. Cholesterol is a fairly simple molecule. We're going to find that it has a particular ring structure. The electrons are all shared. We're going to see that because those electrons are shared, it's hydrophobic. Now, it's true for the different kinds of steroid hormones will modify the ring structure and to some extent the side groups on those steroid hormones. But they're all based around cholesterol, and what they all have in common is they're hydrophobic. The hydrophobic nature of these steroid hormones and also to thyroid hormone dictates a different way. Could you put your phone away, please? The modification here is such that these hydrophobic amino acids are, in fact, these hydrophobic hormones now are modified. Try again. These hydrophobic hormones now have to cross through the lipid bilayer and get into the cytoplasm to bind to receptors. So we have now this big category of difference. That is, there's some hormones that are going to bind to transmembrane proteins. These are going to be charged and big. Those that are going to zip right across the phospholipid bilayer and interact with receptors on the inside of the cell. The other character here, the one we won't really spend very much time on, but again, if you particularly are interested in the immune system, you're particularly interested in parturition, in birth in animals, you get into these modified fatty acids. These are the prostaglandins. Prostaglandins are really important as intracellular kinds of hormones. We're just going to barely touch on them, but they are hydrophobic because they're modified fatty acids. Now, for you to understand what we mean by the endocrine system, you really have to drill down and start talking about the hormone-receptor interaction. Now, we can talk all day long about the various kinds of hormones that are produced and all their characteristics, but what it really boils down to is the interaction between the hormone and the receptor, the specific interaction between the hormone and the receptor. As we work our way through this, we'll find that there are many, many examples where we have the same hormone, the same transmitter, but we get a very different effect by having a different receptor. So it's all about the receptor. Well, to understand that it's all about the receptor means that we have to have this hormone-receptor interaction. Now, there's nothing fancy about this. All we mean by a hormone-receptor interaction is that it's obeying normal physical chemical rules. All we mean is the hormone binds to the receptor, and this receptor-hormone complex sticks together, but can also come apart. When you think about ligand-receptor interactions, whether we're talking about hormones or receptors, or even whether we're talking about hormones and receptors, or neurotransmitters and receptors, or even enzymes and their ligands, in all cases, what you're having is an interaction that is by charge. These are not covalent interactions. They're transient charge interactions. They obey the law of mass action. There's just a question of concentration. And that's what I've tried to portray here, where there's a hormone concentration, receptor concentration, and then a resulting hormone receptor concentration. And this arrow runs in both directions. Well, to try to do this and think about it a little bit in terms of physical chemistry, I'm going to quickly go through this idea of something known as the KD. This is the dissociation constant. And if you take some chemistry, you're going to spend a lot of time on KDs, particularly in biochemistry. And the KD is simply defined as the concentration at which half of the receptors are occupied, the concentration of hormone at which half of the receptors are occupied. That's the KD. This brings up the idea of affinity and specificity. One of the things we're going to find about the KD is that for hormones, the concentrations can be exquisitely low. The KD of some hormone receptor interactions are down around 10 to the minus 9 to 10 to the minus 12th molar. This is nanomolar to picomolar. There's not much of this stuff out here. So if there's not much hormone out here, but we need to have this hormone bound to receptor to get a response, then there must be a large stickiness. And there must be a high affinity for the hormone to the receptor. But you need not only to have the two be sticky, you have to have these hormones and the receptors stick together, but you also have to have the concept of specificity. You don't want to have hormones binding willy-nilly to all bunch of different receptors. They need to be specific. There needs to be a specific interaction between the hormone and its receptor. And this is the idea of fit. So we'll try to bring together the idea that there are charge interactions resulting in stickiness, that's affinity, and there are shape interactions too, which are due to fit, how well these three-dimensional structures can fit together. And this will be just a general idea of how hormones work. And then if there's time, we'll talk about what they do to cells, and we'll see that hormones can affect enzyme activity, they can affect ion channel opening, and they can affect the closing, ion channel opening and closing, or they can affect endo and exocytosis, and finally, gene regulation.